Welcome to the lecture on meiosis. So meiosis is the second type of cell division that we're going to talk about. First we talked about mitosis, which is cell division of somatic cells. And somatic cells are the majority of the cells that make up your body. And meiosis involves the production of reproductive cells, or gametes, which are your sex cells, so your eggs or your sperm. Okay, so mitosis, that's how we grow, repair our body. Meiosis, that's how we produce cells that will allow us to reproduce. So that's why I've drawn you these lovely sperm and egg here on the title page. Alright, real quick, let's talk about some differences between mitosis and meiosis, and we'll look at this again at the end. So mitosis, it consists of a diploid cell, one diploid cell, which we'll call 2N, becoming two diploid cells. Alright, that means they have two full sets of chromosomes. So we started with one set from father, one set from mother, ended with one set from father in this one set from mother in this one set from father in this one set from mother in this one. So the two cells produced are identical to the first one. That's mitosis. Mitosis also only undergoes DNA replication once. DNA replication occurs one times, which is the same for meiosis. And then in mitosis, you only get one cell division. Now again, it also occurs in somatic cells, which we talked about a second ago. I'll put that up here. Somatic cells. Sorry for the cough. Meiosis, on the other hand, starts as a diploid cell and ends as 4 haploid cells, meaning half the original set of chromosomes. So instead of getting, and this is just an example, so instead of getting the two sets like we have over here, this one would only get one set, this one would only get one set, this one would only get one set, and this one would only get one set. So each cell would have only one set of chromosomes. Right? And that's because when we go through reproduction, we want two cells that only have half to come together and make a full set of chromosomes. You wouldn't want to have babies that have 92 chromosomes. Right? So that's why meiosis goes from one cell with diploid, which is 2N, to 4 cells which are 1N, which is haploid. That's haploid. Half of the original set of chromosomes. Only one set of chromosomes. And now they're not necessarily going to be, like for example, this cell wouldn't necessarily get all the ones from the mother. It could be a mixture, but that's just an example to show you how that happens. Again, DNA replication only occurs once in meiosis, but meiosis goes through cell division twice, 2 cell divisions. And that's how we're able to get to that point. This cell right here will divide twice in order to become 4 cells. And we'll see that here shortly. Alright, let's go ahead and take a look at meiosis. So as I said before, meiosis starts with a diploid cell here. Now remember, this cell has gone through, by this point, this is interphase. This is where DNA duplication or replication occurs, right? In the synthesis phase of cell cycle. So DNA replication occurs. So there's 2 copies of the original 2 sets. Right? So technically there's 96 chromosomes in there, or 92 chromosomes in there, but they're copies. Okay, so there's 2 sets of the original chromosomes in there, and they're copies. Once that occurs, we'll go into meiosis 1. Meiosis 1 is the first cell division of meiosis, and that consists again of prophase 1, metaphase 1, anaphase 1, and then we'll have telophase 1 as well. Telophase 1 will be over there on the other side, which we'll see in a minute. Okay, so it's same phases of mitosis, but different things are happening. In meiosis 1, we start here in prophase 1. Again, we have the centrioles that are moving to the opposite ends of the cell, or the opposite poles, and you got their spindle fibers that are coming out. Okay, but notice here in the middle, see how those chromosomes are all lined up? They look like this. So there's one set, right? These are sister chromatids, right? And then here's another set. But then they link together. And that's a terrible way of drawing it. They actually link together and they create homologs or homologous chromosomes. Or another term is tetrad. So you'll see it is homologous chromosomes, or they form what you call a tetrad. And you can see actually where they're, and I'll show you this close up in a minute, but they were actually crossing over. And that's a type of genetic variation that allows us to have diverse children. So we call that crossing over. Okay, and that's a terrible E there. Crossing over. And that's a type of genetic variation. There's 3 types of genetic variations. The first one is crossing over. So what happens is this group lines up next to each other, and these 2 inside chromosomes here will actually exchange DNA. They'll swap sections of their chromosome. And that's called crossing over. They're crossing over some DNA. So you'll end up with, if those were blue and black, you'll end up with now maybe one that looks like this. Oops. Oops, sorry, wrong color. Like that, and another one that looks like this. So now you've swapped up the DNA. The original mother and father set have combined now to create a new type of chromosome. And so that's how we get these new variations in our children. And so that is crossing over. Now I'm going to clear the ink on this page. Now we get to meiosis 1 here. And the tetrads, the homologous chromosomes, let me put crossing over in here. The tetrads line up on, again, that metaphasal plate that we talked about in mitosis. Okay, they line up in the middle of the cell. But instead of the sister chromatids getting pulled apart, the homologous chromosomes, the homologous pairs get pulled apart. So the sister chromatids each go to a separate side. And this is where we get another type of genetic variation called independent assortment. And what that means is these homologous chromosomes, okay, are able to line up however they want. So if we had this one here and this one here, let's say this, let's do this, let's start over here. Let me get different colors here. They could line up like this, and these black ones could go this way, and those that way, and that way, and that way. Or they could line up the other way, and the red could go that way, and the black could go that way. So you can get a variety of variations on which way the chromosomes separate. There's no pattern. They line up however they want. It's independent assortment. There's nothing. It doesn't depend on anything. So again, that's another type of genetic variation. We don't know. There's all sorts of variations that the sperm and egg could get based on crossing over and independent assortment. The only other type of genetic variation, and we won't really talk about it, but that's just random fertilization. That's just reproduction, who you decide to mate with. That's the other type of genetic variation, the different types of combinations of chromosomes that come together during fertilization. So make sure you know those 3 types of genetic variations: crossing over, independent assortment, and random fertilization. Alright, so once these homologs are separated and the sister chromatids go to opposite ends, that's anaphase 1. You can see they start to get pulled to their opposite ends. And then we'll get into telophase. Sorry, we'll come back to that. Then we get into telophase 1, and you finish telophase 1, which is the total separation, right? And they pinch off. That would be the finishing of telophase 1. And it'll look like this. You'll have 2, at this point you actually have 2 haploid cells. These are just haploid here. Haploid cells. At this point they only have one set of chromosomes. Okay, they only have one set of chromosomes. Half of the original set of chromosomes. So they're haploid. But they have a copy. So they have a copy of their set. They only have one set, but they also have a copy of their set. And that copy will end up being dispersed into another cell. Okay. Now let's look back here. This is a more up-close look of crossing over for you. So you can see where the chromosomes line up, they form the tetrad here, and they actually exchange material right there. Okay, and then they look like that and that. And so you can actually see where the crossing over occurred. The exchange of material occurred. And that's crossing over. And that's how we have genetic variation. Alright, now we're here, we're moving into meiosis 2. Now meiosis 2 I'm not going to spend any time on, because once we get to meiosis 2, and it goes right into it, depending on, and there's some variations when we start talking about oogenesis and spermatogenesis, what actually happens, but meiosis 2 is just like mitosis, so it's just like mitosis, so I'm not going to spend any time on it. Just know that phases, but this time we call them prophase 2, metaphase 2, anaphase 2, and telophase 2 would be in here. It doesn't really show it. Okay, the final splitting would be telophase 2 in there. So it's just like mitosis. So meiosis 2 is the same steps as mitosis. Alright, so that is meiosis. 2 cell divisions, 1 DNA replication, diploid to haploid. Again, we get our 4 haploid cells here. 4 haploid cells. Now let's look more specifically at meiosis in humans. Okay, we're going to look at spermatogenesis, so I'm going to put an S there for spermatogenesis, and oogenesis, which is females. So this over here is females, this is males, and they happen a little differently. Males is pretty simple. They don't start it till they're in puberty, and they continue to make millions a day. I'll give you the exact number in a minute, but I believe it's about 2 million a day. So it's very, very substantial. Alright, let's look specifically at what actually happens for spermatogenesis versus oogenesis. So over here we have spermatogenesis. This is the production of sperm in males. It doesn't happen till puberty. And what happens is at puberty, the primary spermatogonium, which is this, and again you can find this in your textbook on page 184, which would be good maybe to follow along while we do this. So if you have your book handy, get that, or if you're drawing this, that's fine. But I'm not going to write out all the names because I don't have enough room. So the primary, or the spermatogonium here, this one right here, okay, the spermatogonium, it divides by mitosis. Okay, it divides by mitosis into what we call the primary spermatocyte, so PS, the primary spermatocyte. The primary spermatocyte then begins meiosis. So we go through meiosis 1, and then we go through meiosis 2, and we create 4 sperm. And those sperm will then be, during fertilization, they can fertilize an egg. Okay, so the model we just saw, that's what happens for spermatogenesis. After meiosis 1, we call these secondary spermatocytes, and these are actually called spermatids, but this is your sperm here at the bottom. So this is your sperm. So your spermatogonium divides by mitosis. So right here is actually mitosis. Because what happens is when it divides by mitosis, so if a cell divides by mitosis, it gets 2 cells, right? One of them will continue through this process. The other one will come back and be a spermatogonium. So that's how males keep that sperm bank going. They're able to just keep refilling the bank in order to keep producing sperm. Okay, females is a little different, so I'm going to clear this ink on this page. So over there, here we have females, and this is oogenesis. And what happens is you have an oogonium here, and during embryonic development, so you're inside your mother still, a female is still inside her mother, she begins to go through oogenesis already, so even before the birth. And what happens is the oogoniums, all the oogoniums they have, divide by mitosis into a primary oocyte. So this is our primary oocyte. And then still during development in the womb, the primary oocyte will go partially into meiosis 1. It'll actually only go into prophase 1, and it'll wait there. It'll go into prophase 1 and wait there. And then when puberty hits and a female starts to menstruate, each month one of these primary oocytes will continue through meiosis 1 and get to this phase right here, where they have a secondary oocyte and a polar body. Now, polar bodies are just going to be absorbed at the end, because what happens is the secondary oocyte takes all the cytoplasm, because it has to be much larger in order to make a nice, nutritious, and sufficient egg. Now, that secondary oocyte, unless it's fertilized, it will not finish meiosis. The polar body will. It'll always finish and create two polar bodies here at the bottom. But the secondary oocyte will only finish if it is fertilized. Okay? So if it gets fertilized, then it will go through meiosis 2, create another polar body, and finally create the egg, which is right there. So the female meiosis is much differently than the male. Happens much differently than the male meiosis in humans. Okay, so make sure you know those key differences, and again, you can find this on page 184, okay, in your textbook. All right, let's review meiosis and mitosis here. I'm going to make this a little bigger. There we go. This right here shows you mitosis and meiosis. As you can see, meiosis goes through two cell divisions. This is cell division 1 here, 1 CD. This is cell division 2 here, 2 CD. Over here, mitosis only goes through one cell division. Now, the second cell division of meiosis is just like mitosis. So, if you can remember that, you'll be fine. Now, remember, you have three types of genetic variation that occur during meiosis. You get crossing over that occurs during prophase 1, crossing over. And you get independent assortment, which occurs during metaphase 1. And then random fertilization is going to occur at the end when you actually have your sperm and egg. Okay. Also remember, meiosis goes from a diploid to 4 haploid cells, and mitosis goes from diploid to 2 diploid cells. Okay. So those are some key characteristics there as well. You've also probably already done the assignment on cellular division, so you've already gone through all these as well. Make sure you review the mitosis lecture as well, and look forward to seeing you in class.